Thank you very much. I just want to tell speaking about stories, two very short stories about Tel Aviv University. And on 88, the Tel Aviv University Business School won the prize of the American Credit Card Association or something like that for being able, two professors, Professor Zahavi and Professor Kalish, and they got the prize of analyzing the data of the credit cards in order to send to the specific customer the right thing. It was just 88. So just imagine what would be the next 20 years following your lecture. And the second story, also Tel Aviv University, that 86, we invited Ohad Naharin, which is the dance choreographer, to look on proteins. Since he is looking on the stability of the body, what will be the stable protein? And it was because he was precise. So, converging between the various areas. So that's just two examples from this university. And last but not least, I want to invite our last speaker. We were talking so much about the brain, speaking about the Brain Connectome. And Professor Yaniv Assaf is from the George Weiss Faculty of Life Sciences and the former head of the Imaging Center. Please.
So first, thank you for staying to the end. And
It's the beginning.
Well, we'll see. So what I will speak with you today is about the connectome. And in order to understand the connectome, we need to understand what we are looking for in the brain. So the brain is less than 2% of our body mass, but such a headache to explore. We know a lot of almost all other organs we have in the body. We know how they work, we know the mechanism, we know how to fix some of them or how to treat some of them if there is a problem, but somehow we are limited when it gets to the brain. We don't know the basic mechanisms of most neurodegenerative diseases. Most of the therapeutic procedures that we have are limited in some way since we don't know how exactly to get there, and it seems that we are facing some limitations in this area. And one of the basic questions that every undergraduate student that comes into the neuroscience programs that we have here in the university and probably in any program in the world, he asks, can we understand better the brain and solve the mysteries of the brain? And it's not that we didn't try to do that. We tried for 100 years of neuroscience to find what are the basic blocks of the brain that are responsible for our cognition and behavior and when everything goes wrong. And a lot of focus and attention was given to the neurons, the basic building blocks of the brain, and the synapses, the basic connection that it makes between the different cells. But just looking at the brain, those two units, which are the most important as shown in textbooks and are the most widely studied in neuroscience. I think 90 or 95% of all research in neuroscience is based on these two building blocks, the neurons and the synapse. There are only 10% of our brain. Our brain is built of other things, other cells that comprise 90% of it. So, and we can argue about the number. If it's not 10, it would be 15, but this is the order of magnitude. So this is a very stunning number. Only 10% are the neurons, and there is 90% of the brain that is something else, other types of cells, that probably responsible for, they are there for a reason. So the question that obviously we should ask ourselves is maybe we search in the wrong location for what's going on there. And this is not the first mistake that we have done in the brain. Usually through the decade, if you look how people try, very famous and very smart people try to explain how the brain works, it's always been done with a mistake. So for example, René Descartes thought that the brain is an hydraulic machine just because this was the most advanced machine that was at this age. Charles Sherrington, who thought of the synapse, thought that the brain will be a Lou machine just because that was the most advanced technology in his era. And later on, in the middle of the previous century, we thought, and we still think, some of us think that the brain is a computer just because this is the most advanced machine that we can manufacture. So looking at the brain as a technology and looking in terms of the biology into 10% of it obviously will not get us to the level that we can solve the mysteries. So what we are missing? So just when you look about how the brain looks, so the brain has the neurons, you can see them in blue, but it has many other types of cells with different names. It doesn't matter what the names are, astrocyte, glial cells of different kinds, etc. But when you just look at it, what you see, what I see, is a connectome. A connectome is a network because each cell of the brain, and this is completely different from any other organ in the body, has the ability to connect with other cells in many locations. So each cell has between 10,000 and 100,000 connections with other cells, and it creates a connectome of its own with other cells, and all together, all the 80 billion cells that we have in our brain create a connectome of each one of us. And this connectome is a map, and the map that represents everything in our body. So what is the connectome? And this is the topic that I'm going to speak about. So the connectome is the mass array of neural connection, not only neural things, everything that we have in the brain. And it doesn't matter. Better?
Yes.
Excellent. And it doesn't matter if it comes from the neurons or from other types of cells. It's the connection that comes from all the different types of cells that we have in the brain that create a very dense networks of connections and components of the tissue. So it's a map, it's a graph that we need to extract somehow. And it spans across different scale. It can be at the level of a single cell that connects with 100,000 other cells, or in the level of the entire tissue. It is important because we believe that if we will be able to map this graph, this network, we will be able to say something about the function and the function of each individual person in that relation. And we believe that this connectome may affect or may represent our cognition and behavior. And some might say that as genes encode our body, we can argue about that maybe the connectome encode ourselves or our mind in that sense. So maybe that was the missing link in history. Instead of looking on one single building block, the synapse on the neuron, look at all of them together and the connections they form, because we do know that the connections are critical to behavior. So how this connectome can be studied? So traditionally, one should study it by just counting the connections, looking at the connections and creating this map. And this can be done with electron microscopy. It was done for this little worm, the C. elegans. It's a very tiny worm with a length of 1 millimeter. And this is an electron micrograph of this worm, and you can expand it and see every little cell and every little piece of organelles that it have. And eventually, when you analyze all the hundreds and thousands of sections that you have for this worm, eventually you can create its connectome, which looks something like that. So it has 302 neurons, and by just exploring the electron microscopy, you can create the connectome. So only for this tiny 1 millimeter worm, the connectome is already so much complicated. So you can imagine what happens in the mammalian brain or in the human brain for that sense. So how can we do it? If we want to explore our connectome, if it can hardly be done to a tiny little worm, how can we do it for neurons with more than 1 billion neurons or 5 billion glial cells, etc.? So an enormous types of cells, endless amount of connections, and this is what we need to explore in order to understand the brain. So the idea that we came here, so this is where we started the project. The idea that we came here is to see or not to see. Obviously, we will not be able to image every little tiny connections that we have in the brain and then get an enormous or endless amount of data that even the biggest computers that we have now will not be able to analyze. So yeah, we don't need to see it, but we need to sense the connections. If we will be able to do somewhat related to a technology called remote sensing, if we will be able to remotely sense the connections and quantify them somehow, and then create a map or a graph of this connection, maybe we will be able to say something about this connectome on the quantification level. All right, so I work with MRI. This is my expertise, and our idea is to use diffusion MRI. It's one method with MRI in order to to remotely sense the connectome or get some information about the connectome and how this is going to work. So first, let me just acknowledge our imaging center, which was, I think, one of the first centers in Israel in terms of neuroimaging, and now has 2 MRI units, one for small animals and one very new for imaging humans. And this center was part of a collaboration that was started with the EU under the FET Open program. It was called the Connect project with Tel Aviv University. We led this project, and with the aim of developing new MRI technology to explore brain connectivity and the connectome itself, with the rationale that in order to understand the connectome of the human brain, we need to do it in vivo. Just taking a brain and dissecting it with electron microscopy will not get us to the level that we need. So we need to do it in vivo, develop a technology that will enable us to do so, and take the advantage of the disadvantage of MRI, because the disadvantage of MRI is that it's not specific. It sees water. This is what MRI measures, water molecules. And this is not specific for any neurobiologist or every biologist. We say water are everywhere. It's not important. We want to look at the proteins. We want to look at the chemicals. But the thing is that if MRI sees water, it sees everything. And in the brain, you need to see everything in order to understand what happens. So this becomes an advantage out of the disadvantages of the methodology. So this was a collaborative effort of 12 different groups from 7 countries, and started in 2009 and ended recently. And we developed a technology that enabled us to take so this is the most basic MRI scan of the brain, so it shows the different structures, but we can take it to a different level with the technology and now jump into every little, maybe it will work, maybe not. And then to jump, well, you could have seen that you can jump into a connection and just go through the connection from one part to another and get a visualization of the connectivity between different parts of the brain. And in the whole brain, it will look something like that. So you have a map of connections that you can extract on different scales. So here you can see the scale of the whole brain, but you can zoom in into different sections and get a more expanded view of the connectivity in each different area, and then create a connectome at different scales of the tissue. So that was the methodology, and there's a lot of other things that were developed there, but this is the basic methodology that we used. And one of the first applications of that is to look how this connectome evolved, because when in the field of biology, when you want to understand if a quantity that you measure, like the connectome, is important, but it's a nice quantity in my view, but some people would say it's not important for anything. One way to understand if it's important is to look how it is evolved, for example, through the mammalian order across mammals. And if it's preserved in the evolution, that means that it is important, because if it's something that was preserved from the mouse brain to the human brain, it means that it is an important quantity. And this is what we have done. So we created the MAMI database. It was in collaboration with Dr. Yossi Yovel from the zoology department, and we have created a very unique database. It's the sole database in the world of this kind. We have scanned 85 and counting different brains of different species across the mammalian orders in order to make a comparison between the different brains, the different connectomes. So this is how the connectome of the human brain, if you will take a professor in neurobiology as a representative of the humankind, if you believe that, but you can also compare it with different kind of animals that present in our database. And this was a challenging project because these brains are different sizes and different shapes, and we need to gather the same amount of information from each of them. So you can imagine that the tiniest animal we have was the trident bat, which weighs 6 grams, compared with the dolphin brain that weighs 150 kilos, and that would have to be scanned at the same relative resolution and kind of information. And that was done. It was very challenging, but was done at the end. And eventually, by taking this connectome for each animal, creating the connectivity matrix. So this is the connectome itself, taking the information from the imaging, taking it out of the three-dimensional, beautiful, colorful space, and getting into a matrix level which the computer can handle, and then doing a graph theory analysis, which I will not go into that. But what was the results of it? The ability of the brain to connect each point on its surface to another point on the surface was preserved through evolution. So the efficiency of the connectivity in our brain was similar to the connectivity in the triton bat tiny little brain, which means first we could reduce the arrogancy of the humankind, which doesn't mean that we are better than other species. We are just the same in terms of our connectivity, and the connectome is a well-preserved structure of the brain, meaning that it is important. It's important for cognition, it's important for function, it's important for behavior. A side effect of this project was that we can create an evolution tree based on this connectome. And the interesting is that it is similar to the phylogenetic tree, for the genetic tree, but with some deviation, meaning that some of our behavior and cognitive, our mammalian cognitive and behavioral abilities causes some deviation from the genomic tree in order to put to create different cognitive related clusters within the evolution tree, which means that, for example, primates are now might be more similar to other species that were in other orders. For example, some of the bats have similar connectomes to humans and other animals as well. The next part of this project was to see, now that we know that we have a method and we know that the connectome is important in terms of evolution, we could go on and look at how dynamic it is. Since we have a method in humans, we can now see how dynamic the connectome is. And the idea here was to scan human subject, to scan their connectome, let them do something, and then they scan the connectome again and see how the connectome have changed due to some cognitive performance. So here we didn't save any money. We gave our subject a Porsche. We sent them to the streets of Monte Carlo with a game called the Need for Speed, where they had to drive a car without seeing the track. So eventually, by paying again and again and again, they could create the track in their mind. They create the map in their mind. And this year, Nobel Prize in Physiology goes to the 3 researchers who actually found the space cells the spatial cells in the hippocampus in the human brain that creates a map eventually. So what was done here? We scanned the connectome with the MRI, then let the subject play with the game for 2 hours, and then scan them back in the MRI. And what we were able to see that for behaviorally first, the subject create the map. They never seen the track, but they were able to draw the track in freehand, so they created the map of the track in their brains, and the time it took them to complete the track reduces, which means that they have learned something. And obviously this should be reflected in their connections, otherwise it wouldn't have been stored in the brain. And we did found that the connectome has changed in certain locations. For example, so this is the hippocampus. The hippocampus is the area where we store information in the brain, and we were able to show that in this area, the connectome or the connectivity pattern was changed dramatically only by 90 minutes of playing a computer game. Think about it the next time you play a computer game. And that was also in correlation with the behavior, etc. But that was not sufficient when we presented it in scientific meetings, because people kept asking, so how long this dynamic connectome is? How long will it be stable? So you change something, will it go away after an hour, after days, after 2 days, a week, a month? How stable it is? So we did the following experiment. So we took subject and asked them to learn the first 50 notes of Forelis. For those of you who don't remember, this is it. Yeah, we can relax a bit for a minute with the music. And what we asked them is just so they didn't know how to play it, and they had to memorize the sequence of the notes. So we scanned them at baseline, then we asked them just to learn the first 50 notes. We scanned them again. So you can see the improvement and the memory. It goes together very well, the music and the lecture, isn't it? So you can see how well they did with respect to the accuracy. And at the end, most of them remembered most of the 50 notes and their location. Then we scanned them again just to see the effect of this learning. And then we gave them another task just to give them a shift in their attention to a different domain, but still with the same procedure. So they had to play Forelis again, but now they had to give attention to the timing of the notes, so where it should be played in order to to create the melody and not just playing the notes. So the timing was important. And timing and accuracy are represented differently in the brain. And then we scan them again at the end. So what we were able to see that the connectome, all right. For the benefit of the lecture, I will stop it. So what we were able to see that just at the accuracy stage, we found that some areas in the brain, those are in the right location, the medial temporal gyrus, the spertum, doesn't matter, but some areas of the brain, their pattern of the connectome has changed. But when we asked the subject to focus on the timing, those changes disappeared or were smaller, but we found changes in other areas that are related to timing as well. So the connectome is a very dynamic phenomenon of the brain. So what is our vision with this connectome or with this quantity of the brain? So in my view, now that we have a method to quantify, this is a very basic property of the brain, the connectome itself, and it's dynamic, because the dynamic of the connectome is our ability to get information from everything we see, from everything we hear, from everything we sense. And it may represent our ability to absorb information. So our vision, and this is what the brain does. The brain just absorbs information, processes, and then output behavior. So this is the process in terms of engineering. This is what it does. So our vision is that we can use now this methodology in order first to better understand cognition, better understand behavior, but also maybe understand what happens when it goes wrong. Because you can imagine that as we use blood tests in order to explore or quantify cholesterol, diabetes, hypertension, things that may lead to heart disease, and we know how to prevent them, because a subject that goes to a doctor and he says you have high cholesterol level, take this pill and it will be okay, it will prevent you from getting a heart attack. Maybe you can do so for the brain, because at the moment a subject says that he has some memory problems when he is 60 years old. But maybe if we'll scan him when he's 30, and we'll see that his connectome and the dynamics of his connectome are not as they should be, we can give them the pill already when he is 30 or 40. Because when we give a subject that has memory problem or dementia when he's 60, we cannot do much to help him, but maybe you can do it before. So this is the vision. So just as blood tests are used to prevent heart disease, for example, we can use MRI scans as the blood test of the brain, and we'll be able to identify abnormal brain plasticity, abnormal connectome dynamics already at young age, in related to dementia, in related to other diseases of the brain, which we don't know almost nothing about at the moment, and use that as preventive medicine procedure. It might take a while until we get that, and eventually to better understand the brain. So this is the vision, and we hope together with this methodology and some other methodologies we develop then. At the end, I want to thank the collaborators and the friends that work, and the students that did the work in the lab. And thank you for staying to the end of this lecture and to the end of this day. Thank you very much.
We are perfect on time. So I want to thank you all for staying with us. Thank the ambassador. Thanks, Professor Ryan, for staying. And thanks to Lea Pais, our head of the research authority, who makes all those projects able to be happen. So thanks all. Thank you. And hope if you want to see the lectures again, just watch the site. Thank you.